This is Mohammed
Yes,
Yunus. It's been a while working with SigmaPlot. So today I will be presenting the new version, SigmaPlot 16, the beta version, what we have right now. I'm going to share my screen. Okay, so we have now the SigmaPlot. This is very classic, the way it was before. We'll have a few new things in it, but before we proceed, SigmaPlot is there for a long time. You probably would know that, over 30 plus years. And then it has been evolved ever since. So we had the first 16-bit, then 32-bit version, and so on and so forth. A lot of things happened during that time. So it is a real coming back to SigmaPlot. On the left side, you can see all open notebooks. So it's called so-called Notebook Manager on the left side. And then you have the tabs on the top. You have the Home tab, Worksheet, Create Graph, Graph Page Analysis, Report, Tools, Macros, and Help. So since version 15, we made some changes, especially in the Help menu. So we put everything exposed. Before, it was way on the right top, and it was kind of, you need to press that tiny arrow to bring it down the menu, and then you would go into it. So it's a kind of hidden before. Now, if you click on Help and then click on About SigmaPlot, then it tells you what version you're running. So this is the SigmaPlot Beta 16 version we are going to present today. And the second important thing is the license status. If I click the license status, then it will launch the license utility, and that's where you actually enter your license. Sometime you are given the product key on the first tab, license activation. There's a product key over here that will be given product key or a V2C file. If you are given the V2C file, you can select that and browse it. So that's a local as compared to the product key. So there is a main difference here. The product key does require an internet connection. So it talks to our server. So once you enter the product key, alphanumeric product key, about 18 to 20 digits long. And once you enter it, you need to apply the license at that time. If your machine is connected to the internet, it talks to a server, it brings back the license and installs it, and that's it. But if you have the V2C file, then it means you may be somewhere out there, or your system would not allow the internet connection to outside world. In that case, you would be given a V2C file. And the V2C file, it's a local, it usually goes as an attachment with your email, and that's what you want to save it on your local machine somewhere in your C drive or on your desktop. Select it, and you need to click browse and then locate that license file like this. You can see right now over here. So this license file, if I select it and then click open, and that's how I can select and apply license. And then on the right, you will see the your product version number, type of the license, if it is a perpetual network site, or the expiration. If it is a time limited license, then it would tell you, and the license manager, this is your machine right now because this is a local license manager. I'm going to close it again. So this is the last on the help menu, on the last two, about SigmaPlot and license status. That's where you want to enter your license. That's very important. People are very confused about it sometimes because they may not know what is the difference between the product key or the license file. They try to open the license file, copy the contents and put it into there, and that does not work. So hopefully that would kind of, I thought, because I get a lot of question about it, so I thought I should bring it up, and that was, to me, it was important to tell you. And the other thing is that you have the product key, feedback. This is what you go onto our website. If you click on the home page, it lets you go onto our website, and that's how you can go, or technical support you need to talk to. Then it will open the support page and you can type your question and so on and so forth. There is in the middle, you can see the sample files. This is very important also. All type of graphs which you produce, most of the graphs are given with the graph and data. And then there's a statistical sample file. That's what we are given. We'll go into that. Transforms, nonlinear regression, macro data. And on the left side, you can see for the guides, you have a user guide, statistics, transform, regression guide. If you click on it, then it will open up the the PDF guide for SigmaPlot. I'm going to close it right now. And then on the left side, you have help menu, you have the contents. You can get into the contents, and then you can type your question, and then you can search for it. And that's how you can get, different titles. And for example, I'm typing Man Whitney here, for example, and I'll go list, and then it will give Man Whitney ransom test. And that's how you can proceed and then go into the help. So this is also very important if you go into that. Then SigmaPlot automation, there's another help for automation tutorial. It is also provided. So if you want to learn a little bit more about SigmaPlot, you want to go to the tutorial, and that's what it is. The tip of the day, it gives you something, pops up. We can close it by default, but that's what pops up once in a while and shows you there's some kind of tip, tell you that, okay, this is what you can do, copy, paste data, whatever you need to do. So let's come back to here, Notebook Manager. So if I were to open a notebook, I can click on the main button, which is the Sigma button, and then I can click New, and then click and create a notebook, and that's what it will open. And on the left side, you can see the name of the notebook for now is by default Notebook 1, Section 1, Data 1. So each notebook would have section, multiple sections also could be possible. And then each section which have the data. Data is the parent object of all further objects. For example, you have the graph page, you have the report, you have any other item that would be the child object of the data. So the all objects should be associated. And the way I can go right click on the section and then click New, and then I can go to report, equation, section, transform. These are the objects. I can go and then open them up if I want to, or create a new one in it. Same way you can open an existing notebook. I just want to go through very simple thing. Let's go and then create a very simple graph, and I just want to tell you how to enter the data manually. So on the top, you can see with a little bit gray numbers on the top, 1, 2, 3, 4. These are actually titles, and we can change the titles. Let's say double click on it, and then I can type X. So this is my X title. This on the left side, it says column 1, and X is the actual title, but it's just to show you where the X title exists is on the column 1. And I can Y here. For example, I'm just using 2 columns. I can select first cell and start entering the data. It will move down the cells as if I keep on clicking enter. And let's say if I can enter some more. Let's say if we can go create a graph and create a graph. Let's go with a bar chart right now. So each, you can see that once you click on the create graph, you have different graphs, but this is the main the graphs type. But if you click on it, then you will get the scatter subgroup, whatever the group you have, and then all kind of other graphs you can do within that scatter. Similarly, line plot, scatter line, and bar chart, and so on and so forth. So let's go with a simple vertical bar chart. We can select it, and then it will open up our graph wizard. We should call it graph wizard. There's another way of opening it up. We can click close, and then you can go create graph here, and then it's a graph wizard here also. So this will also open. Now you can navigate from the first thing. So you will pick up the graph type the way you picked up over here. So now you can pick up in words, vertical bar chart, next, simple bar, next, and you can pick up the type of the data you have, XY pair. If I would have selected the XY already, then it would have selected, but I didn't want to select it just to show you how to select the data. So I pick up the XY pair data. I click on next, and then the way you can pick up the data 2 ways. You can go from this drop down list, data for X drop down list, and then click on X, and then click on Y. Let's say you pick up the wrong data, you can double click on it to deselect it. So this is how it is deselected. Another way of selecting the data, move your cursor on the title, and then you see the title changes, the cursor changes to the up and down with the downward arrow with the big blobs inside. In that case, if you select it, then the data is selected. Similarly, you can move your mouse over here, then it will be selected. One more way, I can double click to deselect it. I can drag the mouse across as like that, so it will be selected. So that's how you can select multiple data at the same time. And if now there is no next button anymore, I can go finish, and I can see the graph. This is a graph title. I can double click on it and make any changes to it, and that's how you can make. Similarly, you can double click over here to changes to your Y label or X axis label. That's how you can change it. These are the tick labels here. And the main jewel of SigmaPlot is the graph properties. If I double click on it, it will bring up the graph properties, and that's where you can change everything, whatever you want to change to. For example, I can go over here, the day under plot, I can go fill, and then I can scroll down, and then maybe I can pick up a color scheme. Any one of those, RD, Heavenly, for example, click OK, and that's how you can see the color scheme is selected, and we can change the colors. It was a very kind of intuitive, very simple way of changing the things. And here you can go graph widths also. It is 60%. You can scroll this bar to the right, so that's how you can go all the way to the right. They will touch each other. That will be 100%. Usually it's about 60%. You can even type it here, by the way. You can type, for example, I can just put 50 or 60. I can highlight, and 60, I can put it in there. And with style, bars, alignment, this also I can do that. You can add also the reference lines. You can have the along the x-axis or y-axis, and then it could be a constant value, whatever that, or upper specification. It could be something, for example, I can go could be a standard deviation 3 times, the third 3 standard deviations, or just a simple standard error value. So that's how you can see the vertical line on the right. So that's on the top of the graph. That's how you can have it, or just uncheck it, then it won't show. I just wanted to show you very little bit how the graph is created. Now you would have noticed that this was the graph page, and as I mentioned it to you, the graph is the child object of the data. Right now, so Torgrim, please remind me if there is a question, because I'm just fully concentrating on my screen, on the SigmaPlot screen at this point.
Yeah, sure, Mohammed. There is one question here. So what is the difference between SigmaPlot and Excel and SPSS?
Okay.
I don't know if you want to take that.
SPSS and SigmaPlot. Excel.
Yeah,
okay. See, the Excel is another application from Microsoft, which has some built-in module solved for statistics. But we're a little bit more. Once I'll show you a few more things in it, then you will see. We used to have a bigger data sheet before, as compared to, I'm not sure how many data now they can bring in. They used to only bring in 65,000 before, but now I think Excel can bring in more data set, but we can go over 1 million or as much you have system resources allowed, the data you can bring in SigmaPlot. And in particular, in SigmaPlot 16, we kind of changed a little bit structure, so you can bring in more data and free up memory, because it used to be everything we used to load into the memory. Now we are swapping on the hard drive. Since people are having the hard drives, they are not spinning hard drives or slow hard drives, they are SSDs, pretty fast hard drives, so it's swapping the data back and forth, and that's how you can read through the data to get more memory free up, so that's how you can use that memory for the other stuff. Just I mean, you won't feel much difference, but internally it is very healthy and helpful for the product. So basically, Excel, you can create the graphs also, but the way you can see over here in SigmaPlot, you can in the graph properties, the one which we called the jewel of SigmaPlot, which at one spot you can do everything, all kind of customization you can do. Excuse me. In the graph properties, as I told you, that just double click on it, and that's how you can get the graph properties. Also, you can go into the home tab, and then here is that graph properties. You can see over here, you can click on it, and that's how you can bring it. The other way of bringing the graph properties and everything about the graph, legend, grids, it lies over here, and you can make changes straight on the graphs. And also, we are more extensive in the statistics. We'll show you in a second, and a few things you won't see in Excel, which we can show in SigmaPlot. Excuse me. Just a second. I'll have a water, sip of water, and then I'll.
That's okay,
Thank you.
Mohammed. Thank you.
Okay, the SPSS is another software which is not very strong in graphs, but it is more statistics. And so we are not comparable to SPSS. We have another comparable product which we call it Systat, which is also produced by us, and that's more comparable to SPSS. And the main difference between Systat and SPSS, Systat comes with all the modules in it. There are only 2 external modules you can purchase separately, but all of the main modules in the regression, classification, regression tree, you name it, a lot of things, those are comparable to SPSS. And SPSS, you have to purchase separate module. For example, for the regression, you have to buy the separate module, and so on and so forth. Some of them, they have the built-in and the base module, but most of them you can purchase it separately. That's what the last information I knew about SPSS. I'll show you something more, and then that's how you would explore a little bit more instead of, I mean, we can keep on hopping. We talk about it, and then we mention it. If I go onto the Analysis tab, and if I click on Advisor over here, you can see the Advisor button, and you see this is Advisor comes up. This is what you don't find in Excel or SPSS. I'm not sure SPSS, they may have it because once upon a time they used to own us. So that's why probably they took it from us, but it was the part of SigmaPlot and SigmaStat way back. I'm talking since I remember, it's been a while. I'm talking about over at least 25 years. So the Advisor, this one does not expect any prerequisites. It does not expect any prior knowledge of statistics you would have. It does give you ask you some questions, and then on the basis of that, the answers, then it will give you some kind of suggestions what to do with your data. So this is for the people, they don't know what to do with their data. It says, what do you need to do? First of all, the first option is to describe your data with basic statistics or summarize your data in a categories. Number 2, compare groups of treatments for significant differences. Number 3, predict, trend, find a correlation or fit a curve. Most of the time, I have noticed that people compare the groups of treatment for significant differences. So that's what I can select for now, and then go to next. Then it asks you the further question by how the data is measured. Your data, is it the numerical values or the ranks? For example, poor, good, excellent. Do you have the ranks? So most of the time people would have the numeric values. Go to next. Do you apply more than one treatment? Sometime yes, sometime no. Let's go with the no for right now. Click next. Okay. So how many groups of treatments are there? Do you have 1 group, 2 group, 3 or more group? or there are two combination of the groups or treatments to consider. Let's say you have 3 or more groups, select it and click finish, and now it will suggest you should run one-way ANOVA, one-way analysis of variance. So that's how it tells you if you have the data. Now, not only that, over here, I'll let you know once we proceed and then do the one-way ANOVA. And actually, let me do that for now for you. I'm going to close it and close this one. Let's go and open the sample data. As I mentioned to you under help, we have statistics samples. If I click on it, and so we have all kind of samples provided, and you can see descriptive statistics, one-way frequency table, so on and so forth. This is one-way data. So one-way data you have, we talked about, remember we have 3 or more groups, so species A, B, and C. This is the data we have. This is what we call it a raw data. The follow-up 3 columns are actually the exact same data, but in a different format. This is the size of the data. The first column has 4 entries, 4, 5, 5, 5, 5, and the mean of each column. So 4.5 the first mean, and then 9.4, and then 4 is the mean of the last column of the species C, and this is standard error of the means. This is how the data format. Another data format, and then the last one we have, which is called the index data. So most of the software, including SPSS, you would find it would ask you for the index data, but we accommodate the actual raw data as well, or you have the other format. So either way you can run it. So let's go and then go to analysis over here, and from this drop down list you can go to different tests. Sorry, again. This is one-way ANOVA. You can see over here. If I click on one-way ANOVA, I can click on run over here to run it. But then there's another thing underneath. It's called options. So you can select the options if should you need to select change the options over here. And I'll talk about it also a little bit more here. This the Shapiro-Wilk Kolmogorov-Smirnov test. I can either click run the test, and it will bring up the dialog for the one-way ANOVA. I can do that over here, or just press the run button here. Once I have selected this from this drop down list, one-way ANOVA, I can click run, and it will bring up the one-way ANOVA. So this is the format I was talking about, raw data, index. This is the mean, size, standard deviation, standard error. This is the one we have standard error one. But let's go with the raw data. I can select it next. And now since we have selected this data already, I can deselect it. So the way I mentioned it to you, I can move my cursor on the title of the first and drag it across. Drag 1, 2, 3, all 3 columns are selected. This is another one selected. I can double, I think. I can do it over again here. Yeah, that's not, this is correct. So you have data 1, 2, and 3, species A, B, and C are selected. I can click finish. And now it is asking me to do the multiple comparison. I can pick up any of the multiple comparison tests over here. Okay, why it is bringing it up? You know what? We are comparing 3 groups, species A, B, and C. In any other software, I'll let you know in a second, for example. So you need to compare here where the difference is. So SigmaPlot actually detected the difference. There is a significant difference somewhere among these 3 groups, but where the difference is, for that you need to narrow it down. That's why you need to run the multiple comparison or the post hoc test, and you can pick up any of those. For example, home SIDAC, I pick it up and then click finish, and that's what I get the report. Now, the thing I was going to talk to you about also, this is, you can see here, it actually tests normality beforehand. So we could go under the options and select which normality test we wanted. For example, we have the Brown Shapiro-Wilk. We have the other one, which is the equal variance test, Brown Forsyth. And follow-up, we have the regular statistics, which means the actual for the column statistics, how many number of entries each column would have if there is any missing value or the mean, standard deviation, sums. And this is the next one over is the ANOVA table. So you have the degrees of freedom, some of the squares, mean some of the squares, F statistics, and then you have the p-value. Here is the conclusion. So now it says there is a statistically difference between among these three. So there is somewhere the differences. Now, where is the difference? It is telling you over here in the multiple comparisons. Is it between B and C? Yes, there is a difference between B and C. B and A, yes, there is one, but there is no difference between A and C. So probably what I've noticed that it went through very smoothly with this one, but let's go and open another data set, and I just want to show you in a second that what I'm going to do over here. This is the similar data. We have the raw data. We can either go index data or raw data, and I can go and then run again. So I can go run this statistics again. And raw data, I can go this time index data just to show you a difference between his index data group is the column 4 depth, and this is the data is the size. So as I mentioned it to you before, this 4 and 5 is the same as 1, 2, 3. This is the raw data. Column 1, 2, 3 is the raw data, but this is the index format of the data. If I click finish, see what happens here. Normality test failed. Do you want to run a one-way ANOVA and ranks? So this is what I mentioned it to you. SigmaPlot does not expect that you would be a statistician before you run it. It is holding your hand. So you could do a mistake and you could run a test which may not be appropriate, even though people may want to go and choose to go with the one-way ANOVA anyway. But this one's suggesting you should rather run a one-way ANOVA on ranks. It is because the normality test so failed. This is what a SigmaPlot does it in the background and holding your hand and wouldn't let you do the mistake. So in this case, it found the normality was failed, so I would go rather run ANOVA on ranks. If I click yes, and now it ran it already, and now it will pick up any test whichever you want to. This is for the multiple comparisons. So there's a little different test. Tukey is the common one, and I can click finish, and that's here is the ANOVA on ranks, which is Kruskus-Wallace ANOVA on ranks. We call it Kruskus-Wallace. And since the normality failed, that's why it suggested you to run the ANOVA on ranks, and that's what your regular statistics, and then which is actually the mean. In this case, we are talking about the median and the percentile value, which is the 25th percentile and 75th percentile values. And now there is a significant difference. Also, you get the conclusion, and then you have the all pairwise comparison, and it is telling you there is a significant difference between 10 meters and 1 meter, but there is no significant difference between 10 and 5 and 5 and 1. So because sometimes if the data is not drawn from a normal population, or the sample is not drawn from a normal population, then you would need, or is it suggested to run ANOVA or ANOVA on ranks, or what's so-called non-parametric ANOVA. The one which we ran it before, the regular ANOVA, that was the one which is here. I can go to this is the parametric test, which is the regular ANOVA, where the normality passed, and that's how you would proceed with it. So any question?
Sorry, Mohammed. We have one about the statistical report you just mentioned. I saw the statistical report mentioned. Is it possible to compute the required? I saw that you had statistical power method. Is it possible to compute the required sample size as well? I don't know if.
To find the sample size?
Yeah.
You're asking how to find the sample size?
For statistical power test.
Power.
I can take
power,
the question.
You see the sample size? Sorry, I didn't.
I can take the
Go
question
ahead.
once more. Yeah, I saw that the statistical report mentions the statistical power. Is it
Yeah, yeah, yeah.
possible to compute the required sample size as well?
Yeah, yeah. Here you can see the sample size. You can see that on the top over here. So I can select that, and then I can go with the t-test, pair t-test, ANOVA, whatever. I can go, and then, for example, I can go ANOVA. Then it asks me a few questions. What is the describe the mean, expected standard deviation, residual, number of groups, desired power, and then you can click, and then that's how you can get a sample size. So there is a sample size calculator over here under same analysis for the t-test, for example, over here. So there is expected difference in the means, for example, whatever is that, 2.3 or 1.5, whatever I can go. And then if it is standard, expected standard deviation, 0.9, for example, desired power, I would have it, for example, I want to have 80% power and alpha is 0.5 to give me the sample size. So desired power must be between, sorry, 0.99, what was that? Oh, yeah, sorry. Because we have over here either sometime I put in a percentage, so I should, instead of 80%, I should say 0.8, and that's how I can get the sample size is 7. You can see on the top it came up. So that's how you can create calculate the sample size.
Yes,
So
thank you.
this is a separate calculator we are given on the top over here for the sample size. All right, so let's go back to again what we were here. So for example, I can go back to advisor again, just to show you. Again, we have, it is asking, describe your data with basic statistics. I could go on that and select it to do basic statistics, but usually go with the compare groups of treatment for significant differences and by numeric values, for example. Next, and if you are applying more than one treatment, let's know, for example, and you have two excuse me, if you have two groups, click next, finish, then you will get a t-test. It is suggesting you the t-test, and you could run it if you have just only the two groups to compare between, so you could go and run the t-test. Let's go back again, and I can go again, compare the groups of treatments for significant differences, numeric values, apply more than one treatment, no for now. For example, 3 or more, there are two combination of the groups of treatments to consider. Finish, it says two-way ANOVA, so you can run two-way ANOVA in that case if you have that kind of data. So you can go and run two-way ANOVA. We have a two-way ANOVA over here, data, and if I don't click on it, it will open up the data. I can, so once I close everything else, still it doesn't close everything over here, but if I, there is, you can see the asterisk on the top, it means you have made some changes to your data, and you can press this button to make save, so everything will be saved. If you don't save it, you can close it, and then it asks you another time whether you want to save it or not. If you want to save it, then excuse me, then it will be saved, otherwise the nothing will be saved to this worksheet or the notebook. So this is the two-way ANOVA data. You can see the two factors. You have a gender, drug, and the reaction. So these are the labels in it. There are two labels, male and female. Drug has A, B. There are two drugs. And then the reaction. So we can go and then run one two-way ANOVA over here. I can click on run and then select same gender, drug, reaction, finish. So it already ran the multiple comparisons. Now it is using the whole, the complete, what you call it, the generalized linear model. It's a gender, drug, interaction term. And I can go finish. I mean, this is the home site, by the way, it's already there, so I can go finish it up, and that's how it will be giving you all the, so this is normal ANOVA. First of all, it ran the normality, it is passed. So that's why it proceeded. And then you have, this is what I was talking about, you have the gender, drug, gender into drug, residual. So that's the whole model. You have a generalized linear model, and there's the power performed. This is all power performed here. And then least square means. So you get more statistics in here, and same thing, all pairwise comparison. Now you're getting everything in here. So this is the between the group, within the group. This is the within the group. You have male, female, and A versus B. This is within also. And you're comparing between now. So within A, female versus male, and so on and so forth. So all kind of the same thing you're getting in here, that there is a significant difference with a conclusion, yes, no, yes, no. So there is a significant difference where, and it tells you in the report. The one thing I didn't talk about it, if you had selected on the right here, you see that create results graph. I selected, then I can pick up any of the results graph. I can create those, and it creates a results graph. For example, group bar chart, it creates it. So you can see that you have the gender versus drug, and then you have the A and B. So that's how it is giving you the graph also. You can go back again and then pick up a different graph, and it has all data is already saved in it. For example, normal probability plot, I can pick it up and then it will give me the normal probability plot. So for all tests, you can go under the, once you have generated the report, you can select this graph, create graph. You can also right click and then you can select the results graph either way, from here, from the menu, or the right clicking over here and then get that results graph. You can also export this report if you want to export it out as a different RTF, different format, text format, HTML format. If you want to export this report out, then it will be exported. Same thing, I didn't show you how to import the data, which is also possible. You could copy and paste the data. This is already built-in data we had. Let's go File, New, and I can click OK over here. In next few minutes, I'll show you what's new in it. I just wanted to show you something about very special features since we brought it up. The question, main question, what is the difference between SPSS and SigmaPlot and or the Excel? So you saw that you can do it things very easily in SigmaPlot as compared to in SPSS. SPSS, you need to know a lot of stuff beforehand. And the one difference here, the main difference, as you saw the report, for example, this one. In SPSS, you have to do everything separately. You have to do normality, equal variance separately, then you need to run your regular statistics, and then you need to run the post-hoc test separately. Everything you need to do it separately. And you need to know the sequence. But SigmaPlot kind of, walk you through, since it doesn't expect the prerequisite, that's why it does for you automatically. Okay, so if you have a worksheet open over here, and I can go and import the data. There is a sample data somewhere. I can go and import, file import. I can select that and import file. I can go into my, this is the main drive where we have SigmaPlot. Your SigmaPlot reside in the C program data, and then there are different folders. So you can see that all the SigmaPlot, they can coexist, but you can run one at a time. So I can go over here, samples, and from here, if I have this Excel file, I could import the Excel file also. This is the file of the type. So I'm going to import something, TXT file, select the contour, click open, and now it has the import text over here. It's delimited already. I can just click import, and this data is imported. So this is how you can import the data. I can simply create a very simple graph. I mean, just to show you, create a graph, a 3D mesh plot if I want to show you. So XYZ triplet, and this is the data I have. I can drag the data again the way I can pick it up. And here's my 3D graph. I can double click on it, and I can go into rotation. I can rotate it. I can change it the way I can make it look upside down, whatever. So I can go and change it. I can also type those here. So all the fields that are open to type, you can enter that, and then you can see the different perspective, different views of the data of the graph. There's another interesting contour, color contour, and this XYZ triplet. You can see my columns are all blue, so they are all pre-selected. So I can click next and just press finish. And here's the graph for our, so you can see these are the depths, these are the crests, the tops, heights, the red, and these blues are the depths, and that's how the contour plot is. Nicely done in SigmaPlot. Okay, so let's go back because we're almost 6:45. I can show you under health. I can go to this macro data set, and I can pick up the different the type of, we have few things we can do, the new ones which we have implemented. Violin plot. So this is the data I have. Violin. There are 3 columns of the data, sample data. I can click on tools menu, and here are the different. So this is the violin newly implemented violin plot. If I select it, then it will bring up the modules dialog. So there are 3 columns already. You can select, and maybe you can just do 1, 2, or all 3. It's up to you. And then you can have a fill color if you have a white, gray, or dark gray, whatever the color you want to do that. It's up to you. For example, this. And let's say the edge color remain the black, and I click OK. Here's that. You can see over here, nicely done. I can zoom it more, 100%, so you can see the violin plot. And not only that, it gives you the data, how it was created, the data behind it. Sometimes you have the large data. For example, I have a very large data here. They're a huge, big data set. You can see it's keep on going. Now, in that case, if you go and run this violin plot, so it's already know it has 3 columns. And there's an option also, you can add the box plot to it. Let's add it and just click OK. It's being given you as a large data. It will take a bit longer to run because it is running some transform in the background. So it will take a bit longer to run it because depending upon the data. So just wanted to show you, if you have large data, so don't worry about it. It will bring it up. So it is running the transform through, evaluating it. Because it has to calculate the kernel density at each and every point. So that's why it is taking a little longer. Here's that. So you can see this graph. Also, you can use this magnifying glass. You can just select that and then view it larger or more larger, only this portion, for example. So you can see this, there is this is the data in the background, and that's the graph. So these are error values, and these dots are actually, if I double click on it, I can show you. These are so we have the plots, box options. So these are the outliers. You can see the outliers. These dots are actually the outliers. We added the box plot in it. So that's how you can see that. Similarly, we have the other also data. Now let's go to the other other graph, which we have new one. This is the census, UK census 2021. There's a two type of census, neutral, count, any other type, and this is the labels. This is the age group, and this is the census of those age group. And I can go and then select this butterfly. And butterfly plot is the one which you're comparing the two groups. One is on the, it's like a butterfly wing. One is the left wing, one is the right wing. And you could have a label. If you don't have the label, you can say no. But if you have the label, you can go ahead. You have to pick it up, the label, from this drop-down list. And if I, keep it everything else, I can also choose the color scheme, and then I can click OK. And here we go. So this is the butterfly plot, the left and right wing. That's how the people compare between the two. Okay. So that's the butterfly plot. There's another thing I want to show you lastly, which is very nicely implemented. Let me go back to the help menu, and let's pick up the graphs here. So this is the one, there are different type of graphs. I just want to pick up the data over here. This data. So it's a data with the error bars. The errors are values are provided in it. I can create graph, and I can go, here is my multiple line and scatter plot. So I'm using the worksheet columns, and then the error calculation is actually coming from here. That's why it is grayed out. I can select from here. If I want to have a column means, then I can pick up the different type of that, but the error calculation. But I can go with the worksheet columns option and next. And this is my X, many Y. This is my X data. This is my Y data, error data. This is another data, and this is my error values. So I can go finish, and here is my graph. And now the people, we noticed the people wanted to have the error bands. So if I double click on it, I can just go into the area fills, and then from here, area fills, and from here, the direction, I can go all the way down and just click band. And see what happened over here? It actually created the error bands. And I can go, and if I want to change the color, I can just double click on it again, and then go the area bands, and then I can go color scheme, different one. Here, I can pick up something else. For example, whatever, heavenly, for example. So, or any other color scheme. This is the same as So this is how you can get the error bands also in it if you want to. So I think most of the things we covered, and we almost have almost 55 minutes. So if there is any question, comments, I can go and answer.
Yeah, thank you for the great presentation, Mohammed. Before we start the questions, I would like to introduce our new product manager and my new colleague in Sweden on the Gothenburg office, Mehmet. Mehmet, can you hear me?
Yes, can you hear me?
Yes, I can hear you.
Great. Thank you, Mohammed, for the presentation, first of all. And I would like to briefly just introduce myself. I've recently joined Alfasoft. It's been like 2 months, and my products are within the quantitative methods realm. And because I have some history with quantitative methods myself, so I am really happy to work with researchers who have that kind of background as well. I really like working with these products. So at the end of these webinars, I will try to contact the attendees. So please keep an eye out on emails from me, and we will be in touch. Thank you very much.
Okay, thank you.
Thank you, Mehmet. Mohammed, there is a question here. I think you already answered the first part of it. Is it possible to run more complex statistical tests like two-way ANOVA? I think you demonstrated that, or regression analysis.
Yeah, regression analysis. You want to do that? We can do it real quick. Yes. Well, regression, I can just come up with real quick data, and then we can do that. We have a few minutes. Why not? Okay. For example, I can enter one. Okay, these are the two simple columns. We have X and Y data. I can title it also X, and this is my Y data. And over here, if I go to analysis, so there is a regression wizard. I can select the regression wizard, and then it gives me the first thing, equation category. You can see the equation category sigmoidal. You can also the standard curves. So linear or 4-parameter logistic. Let's go with this one, logistic. And now it is asking to pick up the data. As I said, that I can pick it up from here, or I can do the other way around, the way I usually do that. I prefer to drag the mouse and then do it like this. It's a personal preferences. And next, and then it fitted the curve. Next, and I can create the report. And under the report, I have a few more things I can do, more statistics. I can pick up the prediction and confidence, and I can select the first available column. Other statistics, there was one person I replied to that AIC, that EK information criterion, the people, they want to see that also sometime. So this is the checkbox, you can do that. And there's more diagnosis. You can explore those also. I can just click apply and OK, and then go next. I can create a new graph. There is a checkbox over here. I can add the 95% confidence. And here you can see the nice bands, 95% confidence, and this is the report. So these are the parameters. You can see the minimum, maximum, EC50 value, hill slope parameter. These are the parameter values. These are the standard errors around them. Analysis of variance, it does it for you in it. And R-square values, here you can see the R-square, 0.99. And you can see the AIC, the one which we talked about. This is what's produced over here, normality test, Wilkes-Barre, and then this is what you have, the Spearman rank correlation, all other statistics, you can find it over here. And the graph you can see over here. Let me enlarge it a little bit to see a little bit better. So here, if I double click on it, if I want to go into the plots, and I don't want to show the prediction interval, these are the red ones, I just uncheck and it will show me only this is the confidence interval, the 95% confidence, you can see that. And if you want to show only either one or both, you can uncheck either one, so it won't show up. Sorry. So this is how you can show up and unshow whatever you want to do that here. And you can also print that. You can export it, as I told you before. You can export the graph also. From here, you can export the graph, different JPEG format or any other file format. You can export TIFF, CMYK, bitmap, meta. So what I did over, as I told you, I can right click and then export graph. And then I can pick up any type of the graph and then just give it a name, and then it'll be it will be exported. So I think